Hello students, I am Dr. V. Sudarshan Rao. So today the name of the module is Microbial Contamination. So as you know, the word itself indicates that micro is small, organism is a living being. So microorganisms are ubiquitous in the nature, that means they are everywhere in the universe. So when food is being processed, these microorganisms can enter anytime from the food production to final consumptions. So in this context, the presence of microorganisms in food becomes very important because it affects both safety and quality of food. So as you also know that many of the foodborne illnesses are due to microbiological contamination of foods. In this context, it becomes very important for us to understand what is the type of microbial contamination that can be there in food and how does it affect the safety and quality of the foods. The learning objectives for this module would be that the type of microbial contamination that you can encounter in food and the factors which affect their growth, that will actually decide how they grow and how fast they grow. And then also the control measures, that how to control this microbial contamination also. These three informations will be actually trying to learn from this module. So microbial contamination is actually the growth of undesirable organisms in food, and they may be bacteria, fungi, virus, yeast and parasites. Is the most important cause of foodborne illness, and foodborne disease can be defined as any disease, usually either infectious or toxic in nature, caused by the agents that enter the body through ingestion of food. So foodborne diseases are a worldwide problem of human suffering and economic costs. So in this context only, it becomes important for us to understand what is the type of microbial contamination that can happen in the food. There are 4 types of microbial contaminations. 1 is the bacteria, 2 is the virus, 3 is the parasites and 4 is the fungi. And based on the type of effect that these microbes do on the food, they can be classified into 2 categories. One is the spoilage microorganisms, the other one is the pathogens. So both of them are very equally important when we are trying to learn on the food safety and quality. Spoilage microorganisms especially affect on the quality of the food, and the pathogens will cause foodborne illness. So decay or decomposition of undesirable nature is usually implied when we are trying to say the spoilage. The spoilage may be due to one or more of these following aspects. The one is the growth and activity of microorganisms, insects sometimes which are actually carries a lot of microorganisms, and sometimes the actions of enzymes of animals or plants, they themselves actually cause spoilage in foods, and physical changes such as freezing and also burning or even drying and pressure, these also will spoil the food. Some of them actually chemical reactions that can happen in food itself. Now we go one by one into each of the microorganisms. The first one is the bacteria. Bacteria are living and single cell organisms. They may be carried by food, water, humans or even insects. So the concern with the bacteria has been they multiply very fast. In fact, if the favorable conditions are available, they multiply into double within 20 minutes. And there are many bacteria which actually produce spores also, and one example could be Clostridium, which is often actually talked in food safety. So there are many genus of bacteria which are involved in spoilage and contamination. I am just trying to quote some of them: Estobacter, Aeromonas, Alkaligenes, Corynebacterium, Clostridium, Micrococcus, Mycobacterium, Pediococcus, Staphylococcus, Streptococcus, Streptomyces, Vibrio, Yersinia. These are all some of the important bacterial genus which are actually found in either in spoilage or in contamination of food. The next one among the microorganisms is the virus. Actually, the viruses are ultramicroscopic living entities. They are the smallest among all the microbial contaminants, and unlike the bacteria, the virus always needs a living cell for its multiplication. Viruses are responsible for many foodborne illnesses like hepatitis A, Norlock and rotavirus. These are the things which actually these viruses cause some foodborne illnesses. So viruses can be transmitted from person to person or from food to people, also from people to food. These contact surfaces can actually transmit some of these viruses. They usually contaminate food through improper food handlers' personal hygiene. If their personal hygiene is very bad, there is likely that they may carry some of these viruses. Bare hand contact also should be avoided with ready to eat food to control foodborne viruses. Even the bare hand, somebody is touching some food which hand is not properly cleaned, also can actually transmit the virus. The third microorganisms which are actually found in foods will be parasites. These are the living organisms that are dependent, as the name itself indicates, it needs a host, either a man, plant or animal, for their survival. Parasites, they take nourishment from the host, and that's how they multiply in the host. So parasites may be transmitted animals to humans, or humans to humans, or even to humans to animals. So there are any number of parasites that have been detected in foods, and these are actually generally coming through the cycle of orophycal route. So some of the very important parasites are Tiniasolium and Giardia, Clistosporidium. So these are some of the parasites that are generally found in the food. So the problem probably more with the sanitation and also sometimes with the non-vegetarian foods. The fourth one is actually the microorganisms which are actually generally concerned in food safety and quality, or the fungi. The fungi are the microscopic single-celled organisms. They actually can be multicellular saprophytic organisms. They are found naturally in air, soil, plants, animals, water and some foods. So molds are multicellular form of fungi which can grow in almost any substance used for food if the conditions are favorable for growth. They spoil food and sometimes cause illness. Some molds produce toxin in food which are known to as mycotoxins. Although mold cells or spores can be killed by heating them, some toxins are not destroyed by normal cooking. That's where the concern is. Some of the normal fungi that we encounter in foods is a mucor, rhizopus, aspergillus, penicillium, trichosicium, geotrichium, neurospora, trichoderma, alternaria, fusarium, endomyces, cladosporium. These are, I just try to quote few of the fungi that are generally found. That list is can be actually accessed in any of the standard notebooks for complete list. The other one, when we are talking about fungus, we are also should be understanding some of the toxins that they are producing. The fungus itself may not be a much concern on food, but the toxins that they produce are very important. That they are called mycotoxins, and most important of them is aflatoxin. That's what we are trying to understand here is that we are not trying to enlist or try to tell you what are all the mycotoxins that are produced in the various fungi, but what we are trying to understand here is which are the things which we are supposed to understand in terms of food safety and quality, where the concerns are much more. Among them is the aflatoxins, which is produced by Aspergillus flavus and Aspergillus species. The four different types of aflatoxins are produced by this particular fungi. They are B1, B2, G1 and G2. So among all of them, B1 is the most toxic and potent carcinogen. Since it is a carcinogen, it causes cancer, the concern is much higher. And these things are mostly associated. These aflatoxins are generally found in groundnuts, cottonseed cake, spices and maize in the context of Indian food products. And wherever the moisture percentage is a little higher, there is a chance that this fungus can grow. Here is that acrotoxin, which is produced by Penicillium and Aspergillus species. As acrotoxin comes in three secondary metabolite forms such as A, B, C. This is the yeast. Yeast are single-celled organism and they gets fermented and converts to alcohol and carbon dioxide. Some of them are actually used as a fermentation process to produce the alcohol. Yeast spoilage may therefore produce a smell or a taste of alcohol. Many times you find that whenever this type of fermentation takes place in food where the spoilage occurs. So now we should try to understand what are the factors responsible for microbial contaminations. So there are two types of factors. One is the intrinsic factors, the other one is extrinsic factors. So now we will go one by one. First we will try to understand what are the intrinsic factors. When I am saying intrinsic factors, it is inherent part of the plant or animal tissues which are referred as the intrinsic factors. This is hydrogen ion concentration. Microorganisms have a minimal, maximal and optimal pH for growth. For their growth they require some pH, and each microorganism has a different type of requirement. Yeast and molds are generally more acid tolerant than bacteria. And depending upon the type of food, the pH also vary. Depending upon the pH, the type of microorganisms also grow. Foods with low pH, that is the acid foods, are generally not spoiled by bacteria and more susceptible spoilage by yeast and mold. A food with low pH tend to be more stable microbiologically than neutral food because the bacteria cannot attack these things. Different types of pH values of different foods. For example, beans is actually the more near to neutral, that is it varies from 4.6 to 6.5. Cabbage is 5.4 to 6 and carrots are 4.9 to 5.2. But now you see in this slide, the fruits are actually acidic mostly. That is the reason why in fruit juices you may find mostly yeast and fungal growth rather than bacterial growth. So for example, apples 2.9 to 3.3. The next important thing in intrinsic factors that we are supposed to understand is the moisture requirement. This is the most important part of the food safety and quality, is that moisture is a very critical point for both the safety and quality. This is required for the growth of the microorganism. That is the reason why we are talking about water. The amount of water required for growth of microorganism varies. Actually, that's the reason why this is called water activity. The water activity is nothing but is a pure water would be 1, and relatively the ideal solute of would be 0.9823. So based on this equation, generally water activity is calculated, and depending upon the water activity, the type of pathogens or even microorganisms can enter the food. Most bacteria grow well in medium with a water activity approaching 1. The most molds differ considerably in optimal water activity, range of water activity for germination for asexual spores. So the minimal water activity for spore germination has been found to be as low as 0.62, but we have seen it is for bacteria you require 1. So you can just see the difference. For as high as 9.3 for other mucor and rhizopus, these type of fungus, they require 0.62 to 0.93. I am trying to give you a few examples. Clostridium botulism is the most studied pathogen. This actually requires 0.97. It's almost near to 1. Similarly, Staphylococcus aureus is 0.86. Here it requires a little bit of salt. So that is the reason why you can just see that this is a little reduced. And whereas you just see Aspergillus, this fungus, you have 0.7. So depending upon the type of water activity that is available with the food, the type of microorganisms will grow in that particular food. Then the third most important intrinsic factor that will affect the growth is that the OR potential, that is oxidation reduction potential of the food. It is determined by the original food and poisoning capacity, the resistance to change in the potential of food, the oxygen tension of the atmosphere about the food, and access which the atmosphere has to the food. This is a little complicated when I am trying to describe this, but it actually means that what is the oxygen available for the organism to actually grow. Based on the ability to use free oxygen, the microorganisms have been classified as aerobic. That means they require oxygen. Without oxygen they can survive. And anaerobic is the organism where it doesn't require much of the oxygen. They can grow in the carbon dioxide also, in the absence of oxygen also. And some of them are facultative. The third type of microorganisms are called facultatives, and they can grow both in anaerobic and also aerobic conditions. Both molds and yeast grow best in aerobic conditions. Bacteria are aerobic, anaerobic or facultative. So bacteria can be all three of them, but yeast and molds are generally aerobic. Nutrient content. So each type of, like humans, each person requires a different nutrition for their well-being. Depending upon the deficiencies or also excesses, he has to actually have a proper diet. Same is the case with the microorganisms. So the nutrient content for each microorganism, a type of microorganism will vary. So how much of that actually will decide which type of nutrient is there in the food will decide what type of microorganism that can grow. The foodborne microorganisms may utilize sugars, alcohols, amino acids as source of energy. Some of the microorganisms are able to utilize complex carbohydrates such as starch, cellulose as source of energy, and some of the microorganisms can use even fat as a source of energy. These compounds are attacked by generally small number of microbes. The things which actually use fat as a source are relatively smaller than the organisms which use sugars and complex carbohydrates. The primary source of nitrogen utilized by heterotrophic microorganisms is amino acids. So the type of microorganisms which are likely to grow in amino acids or even the proteins are different from the carbohydrates. So most of the microorganisms require B vitamins on low quantities. Gram-negative bacteria and molds are able to synthesize most of their requirement. For if they are actually able to synthesize, then they don't require. But in some cases where they cannot synthesize, if it is not there in the food, they don't grow. So this will decide whether the multiplication could be there or not. Fruits have lower B vitamins than meats. Fruits also have low pH and positive oxygen reduction potential, which favor the spoilage of these products by molds rather than the bacteria. So these factors actually influence the growth of molds in fruits. Other intrinsic factors that we are actually have to discuss is the inhibitory substances. Now there are some of the foods like spices that we actually say that they are antimicrobial. Some of them are actually used as a preservatives. They have some inhibitory substance in the food itself. They actually don't allow the microorganisms to grow. So a microorganism growing in a food may produce one or more substances inhibitory or other organisms produce such as acids, alcohols. So sometimes the microorganisms themselves produce or the food itself will have some of the antimicrobial agents because of which microorganisms cannot actually proliferate much. So these things are also considered when you are actually trying to control the microbial contamination in foods. Propanic acid is produced by propanic acid bacteria in Swiss cheese is inhibitory to molds. Alcohol formed in quantity by wine yeast inhibits competence. One organism actually kills the other organism. That's how actually it protects. It actually stabilizes the food, and also storage stability is increased, shelf life is increased. Examples of inhibitors are naturally present are lactoferrin in freshly drawn milk, lysozyme in egg white, and benzoic acid in cranberry. So these are some of the examples where the anti-inhibitory substances are naturally present in your food that itself will not allow any microorganism to grow or allow to grow only some selected microorganisms. The last one in this intrinsic factors is the biological structure. Even the type of the structure of the food itself will actually sometimes will not allow any microorganisms into the food. Take for example, groundnut is there. It is well protected by the shell outside. Unless that cracks, nothing can enter into that. Similarly, you can just see that structures of test of seeds, outer covering of fruits, and shells of nuts and hide of the animals also will not allow you. Shells of the eggs, these are the best examples where the structure of the food itself, like egg, unless the egg is cracked somewhere, the organisms cannot enter into the egg. That's the reason why these are the protection that is actually nature is giving to these foods, which also prevent microorganisms entering into the foods. So that is the reason why damaged fruits, vegetables spoil faster than those not damaged. If spoilage is because it's damaged, the nut is cracked or shell is cracked somewhere, that's the reason why the organism can. Otherwise, if shells are not cracked, you may not find any problem with these things. So also fruits and vegetables, if they are not damaged, they don't spoil faster than those which are damaged. Now we have seen several factors which are actually inbuilt in the food that can actually influence the microorganisms' growth or their control. But now we will see that some factors which are outside the food also, they are called extrinsic factors. These outside factors are like temperature, the temperature where the food is stored, where they are grown or where they are stored, and also the relative humidity, what is the relative humidity in that place, and also gases present. They are all actually considered to be things which are outside the food, and that's the reason why they are called extrinsic factors. Now we'll go one by one. Now we'll just see how temperature affects the microorganisms. Microorganisms grow over a wide range of temperatures, but generally, the most congenial temperature that microorganisms grow, especially bacteria, will be 37 degrees. And if the temperature is in that range, the multiplication is very fast. That means the temperature has a lot of impact on the growth of the microorganism. Based on the growth and survival at different temperatures, they also, even microorganisms can be classified into three. One is the psychotrophs, mesophiles and thermophiles. Okay. The psychotrophs are those things which actually can grow even little lower temperatures, but the mesophiles are generally 20 to 45. Anything little more than that will be called the thermophiles. See, the microorganisms that grow well at below 7 degrees have an optimum temperature between 20 to 30, referred as a psychotroph. This is what we are trying to refer to, is that they grow well at much less, that is less than 7 degrees. That is why you have to just check, some of them actually grow in your refrigerator temperatures, but the optimum is between 20 to 30. These are called psychotrophs. Examples of this can be alkaligens, micrococcus and lactobacillus, pseudomonas, enterococcus. These are few examples. And the organisms that grow well at temperature and cause spoilage at 5 to 7 degrees is a meats, a fish, filter. These are all actually high risk foods which are actually kept in the refrigerators. And the organisms which can grow well at 20 degrees to 45 degrees range, and they are referred as a mesophils and may be held at refrigerator temperatures like Enterococcus fecalis. And the organisms that grow well at more than 45 degrees and the optimum is between 55 to 60 degrees are referred as thermophils. This means these actually can tolerate higher temperatures, and they are Bacillus, Clostridium and Thermoaerobacter. The other extrinsic factors that can actually influence the growth of microorganisms is the relative humidity of the environment in which the food is actually stored. There is a relationship between water activity and relative humidity. Foods with lower water activity are placed in an environment of high relative humidity. Similarly, there is a relationship between relative humidity and temperature that should be kept in mind in selecting proper storage environments for foods. In general, the higher the temperature, the lower is the RH. The foods which undergo surface spoilage from molds, yeast, bacteria should be stored under the conditions of low relative humidity. The last one in this, actually the extrinsic factor, is the presence and the concentration of gases in the environment. Carbon dioxide and oxygen are the two important atmospheric gases used to control microorganisms in foods. As we have seen in the earlier information, that oxygen is very important for some of the microorganisms, and some of them actually grow in the anaerobic conditions. If a microorganism which requires oxygen, if you just control the carbon dioxide in the atmospheric air, we can actually control the aerobic bacteria or aerobic microorganisms. So that is the relation between the presence and the concentration of gases in the environment. There are important gases in the modified atmospheric packaged foods. The other atmospheric gas is ozone, which is having actually, this is another gas that is present in the air, but it's very little quantity, but this is being used in the industry since it has antimicrobial properties and has been tried over a long period of time to extend the shelf life of certain foods. So now we will try to see that what are the sources of microbial contamination in food. Soil and water is the most important thing, because most of the food is either grown or somewhere the water is in touch with the food. So based on this, many of the microorganisms can enter into food. So some of the examples which can come through this bacteria which can come through this soil and water are alkaligens, Bacillus, Escherichia, Shigella, Salmonella, Vibrio. And similarly, air and dust also actually can carry a lot of, they don't multiply in air and dust, but they actually, air and dust will act as carriers for these bacteria. And some of those things which actually come through this is Aeromonas, Bacillus, Clostridium, Micrococcus. These are some of the examples. And plant and plant products also will have different types of microorganisms, and some of them are like, Pseudomonas, Micrococcus, coliforms, yeast and molds, which actually will be there in surfaces of some of these plant and plant products. They actually come through that. And the food utensils, when we are talking about food safety, utensils are used, and with the type of utensils, the contamination that is present in the utensils also actually can contribute to the food which actually we are storing the food. And the food handlers are the another important persons who are actually contaminating the foods. Their personal hygiene becomes very important. Microbial contamination from food handlers is a very critical point for food safety and quality. Animal feeds and hides also will have, because they will carry a lot of microorganisms. The type of pathogens that we see from Indian foods: Listeria monocytogenes, Yersinia, Enterocolitica, Campylobacter, Jejuni, E. coli 0157H7, which are supposed to be emerging pathogens, also have been found from different foods in India. Milk and milk-based sweets, non-vegetarian foods like meat, fish are found to contain more pathogenic bacteria. The contamination levels can vary up to 75%. This of course can vary from depending on the type of food and also depending upon the environment they are actually in. So this will influence the contamination. Then how do we control this microorganism? Now we have seen that how they are growing, what are the sources of contamination. Now we also should understand how to control them. Then how do we control this microorganism? Now we have seen that how they are growing, what are the sources of contamination. Now we also should understand how to control them. Most important part of this one is the storage. It's a very important criteria to control food contamination. Foods which are exposed to insect pests should be discarded as it unfit for human consumption. Any damaged fruits and vegetables should be avoided because the microorganism will gain entry into the food. And foods which are exposed to air also become unfit due to larger number of spoilage bacteria that exist in the air. Of course, it essentially comes in the environment in which the foods are kept. Unhygienic environment, there is a chance that contamination could be high. Food should be always covered if we are talking about at the household level, but at the industry level there should be a proper storage facilities. And the most important thing for this microbial contamination is essentially to try to prevent them. If not, prevention is not absolutely possible, at least to control them to the accepted levels, and this can be understood only by the type of factors that will influence both intrinsic, extrinsic and the sources of contamination. If we are able to understand properly, we will be able to control them properly. And in that context only the HACCP concept has come, and this is mostly used for microbial contamination rather than chemical and physical contamination in reality, but it can be applied in all the cases. To sum up the module on microbial biological contamination, it's very important to understand how important it is to microbial contamination in ensuring food safety and quality. As we have been discussing, the microorganisms can enter into the food chain at any time from food production to the consumption. So in this context, we should be able to understand what are the types of microorganisms that can enter into food, like bacteria, virus, fungi, parasites, and what are the factors which actually influence their growth, either extrinsic, the things which are inside the food itself, and things which are outside, that is like temperature, atmosphere, all these type of things, how they actually influence the growth also has to be understood. And the last and but the least one is actually we should be able to know what are the sources of contamination in these foods. And once finally we have all these informations, there are methods to control. The most important thing in terms of microbiological contamination we generally suggest is prevention is the best, because it's very difficult to control these organisms once they enter into the foods.